Hello.
Hi.
Hi, how are you?
Not too bad. I don't know. I'm doing a carpentry project this week,
Oh.
or it was yesterday, and I'm finished my course, both my courses that I was doing.
Good.
And the exam was Saturday, but I was just auditing, thank goodness.
Oh, yeah, yeah.
They finished the course on Friday, and then the exam was Saturday morning. There were a few complaints.
Yeah.
Yeah. Anyway, so is anybody else going to join us? You don't know.
We'll wait a couple of minutes to see who shows up.
I had a couple of articles about mechanics and embryo development. Maybe I could tell you about them, but I could send them to you later.
Okay. Sounds good.
I just have to find them.
Yeah.
Let's see. This one is called Mechanical Coupling Coordinates the Co-Elongation of Axial and Paraxial Tissues in Avian Embryos.
Okay.
So I haven't really read them, they're just...
Right.
And then there's Mechanical Forces Direct Stem Cell Behavior in Development and Regeneration. That's a 2017 paper.
All right.
Anyway, I can send them to you.
Yeah.
Later.
Yeah, go ahead and send them out up to my email address.
Oh, and Nick's probably on his off week, so he's not going to join us, so...
Okay.
I guess.
Yeah.
Anyway, how are you?
Well, I'm doing okay. Getting ready for the holiday season. And, yeah, I mean, getting that sort of the end of the year, everything in order. You know, reflecting on the year, I guess. I think it's a good exercise to do.
Oh, okay, well, I'm just doing panicked carpentry projects and cleaning, and then I'm supposed to do some baking.
Oh, okay.
Apparently everybody wants cookies for Christmas.
Oh, yeah, yeah.
So I haven't started that.
Oh.
Middle of December. Oh.
Yeah, it does creep up on you. It's like I didn't realize it was the middle of December yet. I mean, I know like on the calendar, but in my mind it's not.
Anyway, I need to get busy. I guess the baking won't take more than 2 days, so I should just do it.
Yeah,
it would be done.
Okay. I don't think anyone else is going to come today. But... Well, anyways, yeah, we can get started on some things. I know people will be watching on YouTube as people can't make it to the regular meeting. So that'll be, you know, you'll be able to follow up on some of these things either in the group Slack or via email or some other means. So I was going to start with a couple, actually our agenda includes a couple things today. I was going to go through an update of a lecture that I gave during DevoWormML. I'm kind of going through those lectures and updating them. This one's on input data. And so we had, last week was the NeurIPS conference. So people who are interested in machine learning and other things like that, that was a major conference in that field. So we had a discussion, I had a discussion with Jesse Parent on Saturday about recapping what was going on at that conference. So it was a pretty long discussion. If you're interested, I can send you a link to those materials. But the input data, I think it'll be interesting both to machine learning people and to people, doing empirical science as well, like collecting data, because it does give like a broader view of data analysis and things like that. The periodicity of the embryo paper, that's something that I am working on trying to get out by the end of the year. We're gonna look at that briefly. And then have some papers that we'll talk about. Oh, there's Krishna. Hello, Krishna.
I'll shut off my audio.
Okay.
I'll try to. There we go. Yeah.
So, yeah, Krishna, we were just talking about the agenda today. We're going to do a... Oh, are you there?
I'm ready. Hi, Susan. Hey, am I audible?
Yeah, you're audible. Okay. Yeah. So we were just talking about, I'm going to do a lecture on input data, which is something that I did a lecture on during DevoWormML, but I'm updating it for 2020. An update on one of the papers that we're working on, and then papers. So let me start. Let me share my screen. I'll start with the input data lecture.
Okay, I'm just going to pop out for a second.
Okay. So this is the input data lecture here. And so this was something that, again, was presented first in 2019, and now we're updating it. So this is input data, or the idea of what comes out, what goes in. So, you've heard of the expression garbage in, garbage out. And that's, if your data is bad, your model output will be bad as well. That applies to statistics as well as machine learning. This is a picture of a system where you have data flowing between variables. And then I've stated this is a Bayesian proposition, if you didn't catch that reference. So I updated the DevoZoo part. The first part I would mention would be that we have a reference for input data for our models. It's called DevoZoo, and this is something that Ujwal has worked on last year to update. And I've worked on it with him. And we've made the input data that we have in our group more accessible to collaborators. You just have all these different files, CSV files that you can download, and you can play with them, do analysis, secondary analysis. And we've curated the data so that it's in reasonable shape for reuse. One of the things about downloading secondary data often is that often the data is too raw, and that is it's not refined enough to be of any practical use. Jesse is here. All right. Hello, Jesse. Hi. Hello? How are you?
Hello, I'm fine. I just joined. It's my first weekly meeting I joined.
Yeah, welcome. Welcome.
Yeah, I actually I joined the community 1 year ago when GSOC 2020 was running, but could not do any meaningful contribution to the community. Actually, I don't have much knowledge about the this kind of work, like how to get the inputs and make some good models to train it. But this year I want to do some meaningful contribution to the community. So that's why I jump in the meeting and want to learn from you.
Oh, well, wonderful. Yes. Welcome. I'm just starting a lecture on input data, so why don't we...
I think that's a pretty good time to join.
Yeah.
Yeah. Great.
Yeah. So Yeah, so yeah, let's go back to the lecture here. Why don't I start over? It was about the second slide, so that's okay. So to recap, this is input data. This is the idea of, you have to put in good data to get a good result out of your model. And this holds true for statistics as well as machine learning or any other deep learning, any other kind of model you want to build. And so we have, this is an update that's something, we had a course in 2019 called DevoWormML, and I'm updating these lectures. So this is where this is coming from. So we have this resource called DevoZoo again. This is where you have a bunch of data sets that we've curated from different sources, a lot of cell tracking data, a lot of lineage tree data and differentiation tree data, and gene expression data, as you can see. And we have them for C. elegans, but we have them for other species as well. And so you can play around with these data sets, and they're well curated so that it makes it easier to understand what you're actually analyzing. But there are a lot of instances of open biological data. There are a lot of sources. ImageJ, for example, has a series of public data sets that you can download. These are data sets that have been captured through microscopy techniques, various microscopy techniques. So they have, bright field microscopy, antibody stains, fluorescent images. So you can get all sorts of images to do a lot of processing and post-processing on them. So, they have data sets that are raw data sets of numbers. They have data sets with images. And I think in these meetings that you've seen a variety of that kind of data. And of course, it's, you might use raw image data for one thing, or raw numeric data for another thing, and combine them. And I'm not going to get too much into that process, but I'm going to talk a little bit about how some of the caveats to watch out for. There are also data sets like the avian vocalization data set here that I'm highlighting. This is someone's work that they put up on Kaggle. So it's a Kaggle data set, and it's just avian vocalizations from California and Nevada. So you can use different, you can combine them maybe with avian vocalizations from other places. You can use it to train a machine learning model. And then there's the SSBD database, which is where we get a lot of our data usually. These are data, a lot of developmental data from various species. A lot of embryos are there. So the two criterion you want to think about when you're looking at open data is it quantifiable? So if you have a bunch of microscopy images, for example, is it quantifiable? Can I turn these images into numbers, and can I do it reliably? And the other criterion is it enough data? So is it enough data to run my model? And so in machine learning, that's a very big constraint because you need a lot of data to train your model. But even in statistical analysis, you have to have a large enough N to make inferences about things. And so you have to always keep those in mind. I'm going to go through these and then I'm going to check the chat. So the 4 V's of data science, we kind of don't think about these too much, but they're very important concepts to understand when you're working with data. The first one is volume. So a volume of data is how much data is available. So this could be both samples or just bytes or megabytes or terabytes or whatever you have in terms of the size of your data set. Now, it doesn't mean that because you have a huge data set that it's really informative data. That's not really, there's no correspondence between the two things necessarily. It's just that you want to make sure that you have enough data that you'll need for your particular problem. But that's always an important issue. Another issue is velocity. How much change over time in terms of movement or process does this capture? So, for example, if you're looking at like a person running a marathon, you don't want to just have a little bit of data of them coming off of the blocks. Or I guess, if they're running sprints, they'd come off of the blocks. But when they're just starting out, and then that's all the data you have, that's not enough data to capture that entire process. We see this a lot with embryos, where you have a little bit of data at the beginning, you might analyze it, but it's harder to say things about what's going on later in the process from those data. So you always have to keep that in mind, that if you want to capture an entire process, you need to have representative data set. And there's a lot of velocity really refers to the amount of change that goes on during that process. So if there's a lot of change that goes on during that process, you're going to miss it if you only have a little bit of data. Then there's variety, which is how much natural variation is captured by your data. So if you have, like, again, a little bit of data representing, oh, there's Mainak. Hello. I might as well check the chat now here, if there's anything in the chat. I don't think there is. Okay. So back to the presentation. So,
Kind of a big data.
what was that?
It's kind of big data. Like all the terms are similar to the big data: velocity, variety,
Yeah.
TVs.
Yeah, that's where it comes from, the big data. It's a kind of a standard thing in big data, but when you're usually, people are, learning about different things, it's something that
Yeah.
kind of gets swept under the rug. So,
Yeah.
yeah.
I
And so,
think
yeah.
it's
So
a
for-
good idea to merge them and
Yeah.
get a good input data set to train our model. And like for the hyperparameter tuning, when you go to tune your hyperparameters to get a good model, then you also need a huge amount of data with good variety and veracity.
Yeah. I'll make a note of that.
Actually, like I know the importance of the data. When like in a hackathon, I needed the data of the corona situation, like how many people are affected with corona. So some similar kind of the thing, and some COPD patient data, but there was lack of data. So I could not train the data. I could not train the model with the data, and I need to create a dummy data set for that, which is pretty bad, and the model was overfitting.
Right. Yeah. We're going to talk a little bit about like the like what we call pseudo data later on. It'll be kind of interesting to see.
More fellows.
Yeah. There's Mayukh. Hello, Mayukh. So let's see. So again, we have variety. So if your data set isn't representative of all the variety in the world, or enough of it that's makes it representative of the world, then that can be a problem. And then veracity is your data reliable before and after transformation. If you have data that is, it's not really something that looks like what's going on in the world, it's not going to be useful. And so all those things, again, they're very useful in machine learning, but also in empirical statistical analysis as well. So how do we know if our open data set is usable? And so these are the criterion that they present in the literature. It has to be available. It has to be what they call usable, which means it has to be documented, it has to have metadata, it has to be credible, it has to be reliable. So you have to know that it represents what's going on in the world. It has to be relevant, so it has to be relevant to the problem. And then presentation quality has to be readable, and the structure has to be there. So that's one of the reasons why we do a lot of annotation, we do a lot of processing of secondary data, is to make it presentable and readable to people. There are a lot of times, if you go to a repository, they'll have standards for submitting your data. If you create data and you submit it to a repository, there are a number of standards you have to follow. Sometimes it might be like a file structure that you have to use. Other times it might be just like different pieces of metadata that need to be there. So this is machine learning training data now. And this is, again, I think a lot of people in the group know the routine here. You have some training data, which we just talked about. You plug it into a model, and then you make a prediction. And of course, it's not as straightforward as it seems. You can have training data that, isn't very good, and it forces your model to underfit or overfit. And so you have to go back and refine the training data. And so, but the training data is an integral part of this. And so we have the MNIST Database, which is a famous benchmark data set. And this allows for model training. Now let's generalize this to input data. So we have a set of various benchmark data sets for training. This is the celebrity face data set. This is the Stanford Cars data set. And then this is the iris data set. I'm not showing the full extent of the variation of the iris data set, but it's a series of these plants, these flowers that are, you can use these data, these image data, decompose the data into features, and then figure out what the shape should look like, what the features of the geometry are, and so forth. Now you look at the cars data set and you look at celebrity faces, and you see some of the 4 V's there. You see that you have, you have variety, of course. You might have velocity in the cars data set, where you may have cars over many years, the different designs of cars. So if I show you a car from 1980 and I show you a car from 2020, those two cars might look stylistically different, but, can you both say that they're both cars? Well, we usually say they're both cars. Hopefully, if you've trained your model on both types of cars, that they can also recognize that those are both types of cars. So those are the kinds of challenges you have in some of these input data sets. The data set should have a lot of variation. They should have, some time variation as well. And then they should also be representative of the phenomena you're trying to measure. So those are some of the things that you want to consider when picking an input data set. And we had another thing in the chat, I believe. Okay, yeah. So Mainak is something interesting to show regarding pseudodata. So, yeah, when we get to the pseudodata part, Mainak, you might want to. I'll let you present that. So that's this. This has to do with data sets for training a machine learning model. And of course, if you're doing a statistical analysis, it's a similar issue. If you want to analyze something, you want to have those 4 V's. You want to take those into account when you make the analysis. And of course, if you do like a standard machine learning is a statistical analysis. But if you step back and you do something like, some sort of classification analysis, or some sort of even like an ANOVA, these same things apply. You want to have a lot of variety. You want to have a large sample, and you want to look at things. You want to make sure you don't violate the assumptions of your test, but you definitely want some variation over time. So this brings us to some more recent example of sort of machine learning and how input data play a role. So this is a new development from this month. This is AlphaFold, and this is something that DeepMind has developed. So people have been trying to solve this problem for years on protein folding, and it's where you try to figure out these proteins, these complex geometries, and how they fold over time. So proteins fold at a rather quick time scale, and their what they call conformational structure makes a huge difference in how they function. And so understanding how the folding process here is very important. But we don't really, it's a very hard problem because it's very computationally intensive, and there's a lot of variation here. You have a lot of parts of the protein, and they fold at different rates, and you have different things going on. So it's a very hard problem, and they've been having contests for years on this. They've usually done this by using supercomputers, using physical models, very large physical models with force fields and things like that, where they've been able to model maybe, a small portion of like a protein's life where it's been, they can model folding, but it's not, it's not a perfect simulation of the process. And so what they've been able to do here with AlphaFold is increase their performance over previous years. And so they've been able to take what they've done with supercomputers and force fields and other types of algorithms that they've used for this problem, and they've applied deep learning to it, and they've been able to get this performance increase. But we have to understand that this progress comes from years of open data input, and open data investment. And so this is someone, Aled Edwards, who says, awesome, but remember, this wasn't by chance. Structural biologists did a couple things. First of all, they set up a data hub 50 years ago called PDB, which contains a lot of the they do like X-ray crystallography on the proteins. So they get a sort of an image of the proteins and their structure. And then, they have the data uploaded to this repository where you have digitized versions of that data. And so then they've also, too, created a culture of data sharing. So everyone who images a protein will upload it to this database. And so it's available for the community. So this is how they've been able to get protein data that's, diverse and really kind of understand what proteins are doing. And then they focused on data quality. So the PDB hub, they enforce static standards, quality standards for input data. And then they ran competitions to benchmark prediction methods, which are these competitions you saw earlier, which means that, it's forcing people to innovate and forcing people to be sort of adhere to community standards. And then 5 contributed over 10,000 new structures to this exact end. So this wasn't just like an advance in deep learning by itself. The input data played a huge role in driving this innovation. So let me look at the chat again. We have another comment. Yeah, AlphaFold 2 is really big right now. So yeah, I think that's a good example, but it's also a good example too of input data and how it's really enabling a lot of these kind of advances. So these are pre-trained models. I made this slide last year, so I know we've done a lot of work on pre-trained models in the group since then. Talk about pre-trained models as an architecture that is validated by a benchmark data set to solve a similar problem. So this is common across deep learning and nonlinear programming, where they use pre-trained models for different ends. In some cases, DeepNet or Mask RCNN, the model is generalizable, can be generalizable to a host of problems, but not necessarily specific to a problem domain. So we have this deep learning model, Zoo of ImageNet, ResNet, Inception, and DGG. And here's some of the references. And then we talk about pre-trained models. They also allow for clearly defined features and classes to be built from a data of a specific type. So, scenes or faces or cars or tomatoes. There's our article highlighting 10 pre-trained models with data sets. This is from a data analytics blog. And then overall pre-trained models also allow for faster training, often with superior performance. But this may not always be the case. So we should, and then in this article, they tell you to approach pre-trained models with caution. And so it's not all, wonderful stuff in pre-trained model land. There are some caveats that you have to consider. And a lot of those have to do with input data and training. So pre-trained models use input data to, sort of prime the main model for performance. But if you build your pre-trained model on bad data, of course, that can be a problem. We also talk about data augmentation. So data augmentation is a way to take a data set that doesn't have very much variety and add variety to it. And so we've done some work on data augmentation in the group with embryos. Examples are where you take a dog, say, and you take this image and you stretch it and you put it at you shift it to different orientations, and you train them, and you train your model, your pre-trained model, or your deep learning, or your your machine learning, deep learning model on these different variations. And so it's teaching the dog to, or it's teaching the model to look at the dog from different perspectives, not just from like a vertical perspective as you would see on the left. Another example is where you take, like, say, like a giraffe, and you augment the background or the shading, so it doesn't expect to see a giraffe as like this, just the static giraffe like this. It can see it from different orientations. If the sky is hazy in the image, it can pick the giraffe out. Or you can create masks, which actually allow you to pick out the general outline of the feature. So, instead of looking at something that maybe looks like a giraffe, something that's spotted, you might have another animal in nature with this kind of coloration pattern, and it might identify it as a giraffe. You can use this kind of mask to break it down into these geometric contours, and that also gives the model information. So augmenting the input data is important as well. So you can do things also like upsampling and downsampling. You also have to take into consideration balancing your training set or your input data set into different classes, so that each class is represented by a similar number of samples. You might downsample to get resolution, to match the resolution of other sets, or you might upsample. So say if you have one class where you only have a couple instances, but other classes where you have a lot of instances, you can do these sort of strategies to normalize for that. And so, this is one thing you have to keep in mind when you have input data, that it's not always neat and even in terms of the classes that you're building, especially when you put data sets together from different places. You might have some classes that are overrepresented, some that are underrepresented. And so these are strategies to normalize all of that. So now we're getting into synthetic and pseudo data, and so this is something that Mainak had mentioned he had an example of. So synthetic or pseudo data is a model of data that generates something not found in the original measurement, or something that's not directly measurable. And so an example would be interpolation between samples, dynamical modeling of a hypothetical regulatory process. So like in systems biology, we'll often use, we'll often have like some model that we use for regulation, but we only have some of the genes measured. And so to measure some other things, we might create like some stochastic process. So we'll generate some data representing the stochastic process. We'll simulate the data, and then maybe use some of the things we know about the intermediate processes to modify that stochastic simulation of the data. So you can do things like resample real data through jackknifing and bootstrapping. You can do this to create like a different variable or something that's approximating what you want to measure. You can also use small sample inference, which is distribution modeling. So you can take like something you only have a couple of samples on, and you can build a distribution. So you can say, well, I only have a couple samples of this thing, but I assume that it's coming from a Gaussian distribution. So I'll build a Gaussian distribution around the parameters that I measure from my few samples, and then sample from that distribution and generate new points. And I can assume that this is the way that the rest of the data is distributed. And so it's a good heuristic for my data that I want to understand. And finally, you have things called data-dependent priors. So you can use Bayesian inference to generate priors for, say, like a Bayesian model. So this is where you observe data, you build a prior, and then you try to predict, something from that prior. So I'm not going to get into Bayesian inference, but that's another way to go. So how do you make a pseudo data set? So the first step is to create archetypical labels, or like figure out what you want to create in terms of a variable. And so what do you want to measure? And so you want to create these and be clear about what you're trying to measure. Then you use a distribution or a statistical distribution to create a series of plausible values based on estimates. So you might have like a mean and a standard deviation, and from that you can you can build like some sort of distribution. You can build a Gaussian distribution. You can build a exponential distribution of some type, but you're going to get a larger number of values, and those values will represent that process, but it won't be the real measurements. So you want to make that clear. And then you want to sample the ways to sample these values in ways that test hypotheses and allow for variation to be represented. So you want to sample, and sometimes you want to resample or jackknife your data so that you can, try to understand, the variation that might exist there, or to create, data in ways that are more, amenable to what you're actually trying to measure. But you also want to be able to, test hypotheses with it, or to plug it into a machine learning model. And, there are ways, like we mentioned before, you can have underfitting and overfitting if you don't do this process correctly. So it's basically 3 steps. And, like here's an example of pseudo-labeling for something, where someone's doing semi-supervised learning, which is where you have, you have a lot of unlabeled data, but you have some labeled data, and you want to kind of augment your unlabeled data. So you use a process of semi-supervised learning to do this. So you create labels from unlabeled data. So you're just guessing based on features in the unlabeled data set. And then where you can use a more formal set of labels to weed out false positives. So you can take the labeled data, you put it into a model, and then you train the model with labeled data, and then you use the trained model to predict labels for the unlabeled data. Then this results in something called pseudo-labeled data. And then you retrain the model with the pseudo-labeled data, which is where you have this process of labeling unlabeled data from a sort of an initial pass. And then that generates labeled data for you from the model. And so this was covered in a blog post. This is just one way to do this. But really what you're doing is assigning categories to things that don't have categories. You're doing this in the same way you might try to identify features in a data set, or do some sort of prediction from labels. And so that's all. Actually, now Mainak had an example of pseudo data that he was going to share with us. I can't.
Can you hear me?
A little bit.
Yeah. Hello?
Yeah, I can hear you now.
Is it okay now?
Yeah.
So can you share my screen like you were speaking about pseudo data? Like,
Oh yeah, let me stop presenting from it. All right, go ahead.
Yeah, okay. Yeah, can you see the screen?
Yeah, it's coming up now.
Yeah, so you were talking about pseudo data, right?
Yeah.
So like you were speaking about synthetic and pseudo data, and one way to generate pseudo data is like using GANs. So I guess you know about GANs, right? So,
Yeah.
And so here what you all can see on the screen is some samples from the MNIST dataset. Like these are some handwritten digits which is widely used to train models. So let's say you wanted to train a GAN to generate some more handwritten digits, So like if you train just a GAN, so if you train a GAN, you get outputs like this, these are not really digits. Like these are some, they have features like digits, but they are not really digits, right? Like some of them are 3, some of them are 1s, but the rest are just area digits, right? So like one drawback of using this GAN approach is that it does not consider the labels, Like when you are training the model, it just takes into consideration whether or not it's a digit, but it does not consider which category it's on. So it doesn't know if it's a 1 or if it's a 2 or if it's a 3. It just gives out samples, that's all. So, but this problem can be solved using something called conditional GANs. It's like really new. This is like really new. So I'll show you another sample. Yeah. So these samples that you see here. Can you see the samples? I don't know.
Yeah.
Yeah. So these samples that you see here, they are generated using a conditional GAN. So the difference here is that we are using a very similar framework. Like we are using two networks, the generator and the discriminator. But in this case, we are also using the labels. Like the greens are the labels. You might see these. Like
Yeah.
so, what we are doing is that with the images, I'll show you the notebook. Can you see the notebook?
Yeah. Yeah.
Yeah. So we have got two. So like any other GAN framework, we have got two networks: the generator and the discriminator. So what the generator is doing is that it takes in a random noise vector and also the label. Like suppose for this image the label would be 6. It takes in the label embedding, which is 6, and it also takes in some random noise, and then it keeps upsampling through the convolutional layers and it gives out the output of 6. This is the generator. This is like in simple terms, like you guys can consider the generator network as an art forger, which is trying to create fake data that the discriminator will try to find, like if it's fake or not. The discriminator is like the cop and the generator is like the forger. This forger is trying to sharpen its skills by making more and more realistic digits, and this discriminator is trying to sharpen its skills by training on the fake and the real samples and trying to discriminate between the real and the fake samples. So what the discriminator does is that suppose the generator has generated is 6, right? So this will take the input of the 6 and then it will pass it through these convolution layers, and then it will also take in the label embedding, and then it will output the probability of the input image being fake or not. So as we saw, like in the traditional GAN framework, the labels are not considered, and we got some alien looking digits like which don't really make any sense. But like if we consider the labels, then we get way better results. Like I'll show you some results. Yeah. So these are some results that are output from the trained model. So these look quite good compared to the traditional framework, right?
Yeah.
So you all might be able to see this. Can you see this here?
Yeah. Yeah. It's.
Yeah. So it's there, right?
Yeah. It is.
Yeah. So as I had mentioned, like in the generator network, we are taking in some random noise. So this random noise in this case it's a vector of length 100. So like a vector of length 100 is nothing but a point in a 100-dimensional space, right?
Mm-hmm.
So like in that 100-dimensional space, what happens if we move around in that 100-dimensional space? Like this random noise, let's say this random noise vector keeps moving around between two extremes. Like for one sample of random noise, it will generate only one type of digit. Like it will look exactly the same if we give the same label and the same noise. But for let's say if the noise is slightly different on a separate point, then it will be something else, right? So like as you might see, like this case is interpolating between 2 points, right? So like what I basically did is that like I took 2 points in that 100-dimensional space, and I took some points in the middle, like I took some interpolation points between the two, and then I generated samples in between them. Like you can see, like they're moving out.
Yeah.
Like they're morphing into one another, right?
Yeah.
Like it's hitting one extreme and then it's looping back. So I guess you were speaking about generating data. So this is another way you can generate data, like not just upsampling, but like GANs can also be a really, really reliable form of generating data, you know. So I guess GANs can do wonders in this situation, and that's all.
But I think GANs are computationally intensive sometimes.
Yeah, absolutely. Yeah, absolutely. Like training is they take a lot of time. So what I do is that I usually use GPUs. As we are moving forward in time, GPUs are getting cheaper and cheaper. It's like every day, like everything that I've done is on Google Colab, so it's free, I guess. So it's something really accessible now. It wasn't a couple years ago, but now it is actually. So
Yeah.
that's something really good.
Yeah, but I think if for like creating some labeling data for fake labeling data, if we can find some other easy ways to do it, I think that's a better way to try with that easy one first, and after that we can go to the GANs or something
Yeah,
like this. But because
yeah,
like it's kind of lot of like
yeah. There's a lot of math and
yeah,
there's a lot of computation involved. But like if you
yeah,
can do it, you get the best results using apps.
yeah,
And
yeah, that's right. But
like if
like
speed is not the factor, if speed is not the factor, then I guess GANs is the best way to go. Like you get the most accurate results if you go with GANs. And what I was trying to emphasize on is the conditional GANs. That's what like GANs have been there now for a long time. Like the DevoLearn library, like I saw like in a sub module of the library, we have got a part where we can generate embryo images, right?
Yeah.
But yeah, but like the classes of like the output that we are getting from the library, the classes are not specified. Like we can't get an output for a specific class. It's just giving out a random output of a random class, a generated output of a random class. So I guess conditional GANs can really be helpful in this case. Like suppose the user wants a generated image of an embryo of a certain class. So that's how conditional GANs can really help.
Yeah.
and and one question label will is label is converted to an array when it is
Yeah, just like anything else in a PyTorch model, anything is should be converted to tensors. So this label is actually one hot encoded into a tensor.
yeah, I think about it like
Yeah,
one
yeah,
one
and yeah, exactly. Like anything in PyTorch, you have to convert to tensors. So that's what.
can
And
you share the notebook in the Slack channel so we can take a look.
Yeah.
yeah, absolutely.
Yeah. Post it in the chat as well. Just the link.
Yeah,
So yeah, I think that's a good thing for people to look over and try. So yeah, I like that. I didn't even think about those sorts of things. But yeah, there are a lot of ways to create pseudo data, and this is of course one. If you're doing this for just what I'm showing you is sort of a very general thing, more maybe oriented towards statistical analysis. But yeah, there are a lot of ways to generate pseudo data, and of course, we're trying to. The whole reason we're doing this is we're trying to get maybe more input data than we otherwise would have. So we're trying to simulate the input data a little bit more, maybe augment it or do other things. So thank you, Mainak, for that. That's very helpful. And so the next thing I want to talk about was, now we're getting into sort of some of these things about what the sort of the pitfalls of small data sets are. And so one of the things we find is that there's this paper called Lessons for Artificial Intelligence from the Study of Natural Stupidity. And they find that, when you have data, sometimes even when you have enough data, but it's sort of biased in certain ways that you're not aware of, your machine learning algorithms can replicate human biases that exist. So, if you have small and incomplete data sets, and sometimes it's not so much that your data set is incomplete, but it has difficulty interpreting rare events. So like, for example, if there's a rare event in the world, something that doesn't happen very often, if you train your data set on a lot of normalized data, it's not going to pick it up. So like in the MNIST example, say there are certain people who write their threes backwards, and that's a 3, and this is something, how people sometimes cross their 7, sometimes they don't. If you don't train it on that variant, and it's a rare variant in the population, but it exists, then it won't be able to interpret that 3. So that's one thing to consider. Another thing is the ability, machine algorithms' ability to learn from decisions is based on choice contingent feedback. So, there are all these things in human reasoning about risk aversion and taking consequences from false positives and false negatives and generalizing it. And so these are all things that machine learning algorithms can suffer from as well. Finally, there's something called biased inference and evaluation. And so this is what they call the attentional learning trap. That's where the better choice is ignored because it's not obvious due to saliency or recency biases. So in other words, if something isn't really salient or something isn't recent, then it can be ignored, even if it's a better choice. And so, all these things we have to consider. They call it natural stupidity because I think there's been an assumption that machine learning algorithms are sort of a triumph on human decision making and some of the errors that we, from memory and decision making, but that's not exactly the case. And so this paper is kind of an interesting take on that. But to think more about this idea of rare events, there's a lot of literature in machine learning and in human cognition that kind of deals with this. One of them is the out of distribution generalization. So there's been a lot of work in the machine learning literature on out of distribution, generalizing from something that's outside of a distribution. And so these would be these rare events I just mentioned. Usually we will train our models on distributions that are IID, which are independently distributed events that, they're idealized, so they exist in that, and maybe in a Gaussian. But of course the Gaussians also have tails, and so those tails aren't very well sampled. And so if you have something that exists in a tail, you're going to, misinterpret it. So what they've done is they've trained models purposefully on these out of distribution events and trained it to generalize to those events as well. So this is the example you see where, these are sort of the out of distribution areas that are sparsely sampled. But if you make a point to sample those areas, you can improve your performance on those rare events. Another thing is burstiness. So we think of periodic events, and we think of things, but things in the world aren't distributed periodically. In other words, we think of things as happening sort of, maybe every 5 units or 10 units of time, and so in the natural world, you have a lot of different types of distributions. Some of them are Poisson distributed, some of them are bursty, and you can see the differences in how they occur. And so if you train your model on sort of periodic set of events, you're going to miss a lot of this natural variation in how things happen. And so, not to get into the distributions here, but these are events that are clustered in time, but there's no meaning to the clustering. It's just that that's the way the process operates. And so, that's something to think about as well. And of course, rare events, and there's something called extreme value theory, which if you're interested in rare events, you can understand. This is more of a statistical aside, but I would encourage you to think more about these sorts of things. And then, when do small data sets work best? Sometimes you don't want to do all this processing of your input data. Maybe you just want to use a small data set. And so there's this paper, Less Data is More, Why Small Data Sets Hold the Key to the Future of Artificial Intelligence. And they use an example from natural language learning. And they use small data sets as a means for concept learning. And they say that in small data sets, sometimes you can use human-machine collaboration to do things like classification and verification of the data. And so, sometimes you'll have a small data set and you'll find something in the data using an algorithm, and then you'll verify it with a person. So you're not trying to generalize with the machine, you're just trying to create, maybe extract features or find an interesting pattern, and then the human operator would verify that and say it's something that's interesting or not. And this is something that allows for user selection when users are selecting on these things presented by algorithms that are mining data, small data sets. It allows them to find the correct answer in a small amount of sort of a, different iterations of the thing. So like, if it presents me with a bunch of options, I can select those options, and then I go to the next round and I select the ones that it comes up with in the next round. And then after about 2 to 3 trials of that, you can converge to a correct answer. In the original, going back to biology, consider the following spherical systems. Now we think of data, we've talked about the MNIST dataset and we've talked about the FACES dataset, but think about something like this. So there's a lot of input data here, potentially. There's, this shape data, but we also have dynamical data. We have data on cell divisions. We have data on deformations of individual cells. We have movement data. We have a lot of things going on. And these are 2 different embryos, and this is just embryogenesis. So there's a lot of, physical interactions. There's a lot of data that we can extract from this and use as input data to our models. And so there are a couple questions for application specifically to developmental biology. And the first one is, what does a grid training set look like? What properties should it have? So should it be, based on microscopy? Should it be based on genetic, data? Should it be based on electrophysiological data? Are pre-trained models adequate? And we kind of know a little bit about that from our work last summer. They're adequate, but we still want to do more work on this. Do we need biologically specific models or multi-scale models? And then finally, what are the semantic aspects of biological data? Are they just labels, or are they annotations of function, or are they shapes, like the shapes I showed you there, where you have spheres or blobs? Are movements semantic, like when cells migrate, or functions? If cells have a different function, even if it's just kind of watching it like, contract or watching it move, is that a semantic thing? And so those are all interesting questions. And then I would finish with this. This is what we have now. This is the state of the art in the project DevoLearn. This is where we have a pre-trained model, DevoLearn 0.2. This is optimized to segment and analyze high-resolution microscopy images. This is some information on DevoLearn, but we also have a platform DevoLearn, which is this initiative to sort of bring together all of the machine learning tools that we have in the group and allow people to learn around that library of things. So we have secondary data, we have these analytical tools, and then we have an educational collection, like data. I think a couple of you have contributed to the data science tutorials, and so we use that as a means to facilitate a lot of this type of thinking in the group. So that's all I have on that, and I will probably be updating it again. I want to put some of your input in, and I'm going to provide authorship. We have a separate stub for this on GitHub, so I'll be communicating with you about this in the future. I think this is a good topic for people to learn about, and I don't really see it too often in other areas of we just had NeurIPS last week, and I don't think anyone talked too much about input data at the conference, but it's definitely an important issue to talk about. So I wanted to present on a couple other things today. And if you have to leave at the top of the hour, that's okay. I'm just going to keep going until we get some of these other items off the board. Thank you for your attention on that. That'll be useful for another round of revisions on that. The first thing I wanted to mention is this periodicity in the Embryo paper. And so this is a paper we've mentioned for a number of months. We've been talking about working on this. This was a paper that we tried to do last year, and it didn't really take off. We're going to be submitting this to a special issue of a journal by the end of the year, so the end of the calendar year. So that's in about 2 weeks. And I'm working on the draft now. It's basically this idea that in embryos you have this sort of these temporal features, I'm calling them, and it's basically this one of the ideas I showed in the slides of how things are distributed in time. And so there's this idea of looking at cell divisions and how often they occur in the process of building an embryo. And it turns out that they're very bursty and they have a lot of interesting properties. And so this paper is going to go through some of those properties. We use two different animal models. We use the C. elegans, which is what our group is named after, the roundworm. And then we have Danio, which is a zebrafish, which is a model organism. It's a fish, obviously. And it has a different type of embryogenesis, but we can make comparisons between the two. And then we use simulated data, which is a very simple method, but it's informative with respect to some of these distribution-based approaches. And so the introduction goes over why this might be important, how it ties into the literature. Then I talk a little bit about some of the theoretical underpinnings of it. I have some methods I have to flesh out. And then we have some figures. We have some data from C. elegans where you have these birth times distributed in, bins. So like every 5 minutes of development, we have a number of these division events. And so that represents a bin in this graph. And so you can see that these are things that happen in the embryo. These are things that happen post-embryonically. And then we have this interval analysis where we take the intervals between these and plot them out and try to figure out what the distribution of those intervals look like. And then we go back to zebrafish and we look at similar things going on in zebrafish. We have this time period, which are 2 developmental periods. The closer inspection of the first developmental period. It looks like there's a lot of there's a sort of a regular pulsing here, but it breaks down later on in development. And so this zygote stage, it's more regular than the cleavage stage. So we kind of focus in on that. Then we do another analysis of the intervals, and then kind of get into the synthetic embryo where we're generating a bunch of division times based on different distributions that we assume that these divisions conform to. This is a uniform distribution of cell divisions versus some of the other types of cell divisions using other types of distributions. We have to change that label. But this is basically, and then this is basically the results. I'm going to write up about that. Then the discussion, which is just kind of interpreting the data, then the references. So it's pretty short right now. It needs more work. I'm going to be working on it. If you're interested in collaborating on this, I can send you a link to this doc, and you can read it over, give your comments. I'll probably send it out for comments in Slack before I submit it. So that's, and if you have an opportunity for authorship, if you can make comments or maybe make some additions or changes, then that's something that you can also, that's an opportunity for authorship. I'm pretty open with respect to authorship on these sorts of things. So that's that paper. Hopefully we can get that done by the end of the year. And then we have some papers. So usually in these group meetings, we go over a couple of papers that were interesting in the last week or so, and we kind of review them very quickly just to give people a taste of what's out in the literature. And if you're interested in following up on this, I can give you a link to this folder. So I'll put it in the chat. And okay, yeah, we had some Krishna wants to present something, and Jesse says, if you need any review or copy editing, let me know. Yeah, we'll be talking about that. So Krishna, did you want to present something at some point, or?
Hi. You can present your papers. I'll present afterwards.
Okay. Yeah. Let me go through the papers real quick. and then we'll do that. So one of the things, I found this last night, I was reading this. This is a blog post by someone called James Sommers, and he says, I should have loved biology. And it's an interesting take. He's a computer scientist, but he's not really that well-versed in biology. And he's trying to understand, he wants to really understand more about biology. So there's a lot of really exciting stuff going on. So he's talking here about the state of sort of getting sort of into biology from a very, baseline position, I guess. We probably took high school biology, but there's just, when you try to read an academic paper, it's almost impossible to really kind of get through a lot of the jargon. So he says that he's starting a magazine assignment to answer some questions about SARS-CoV-2, which is, of course, a coronavirus, and the immune system. And he reads an academic paper where they have a paragraph like this. So, they have a lot of different abbreviations in here. They have a lot of jargon terms, total reads mapping, more than 300 times coverage across the 30 kb genome. What does that mean to someone who doesn't really know a lot about biology or genomics? And so just kind of, the idea is there's a lot there that sits in the way of people really understanding what's going on. And so, a lot of this is bouncing back and forth between Wikipedia and the article. But how can you make this process easier? And so, so he says, but biology, like computing, has a bottom, and the bottom is not abstract, it's physical. It's shapes bumping into each other. In fact, the great revolution of 20th century molecular biology was the coupling of structure to function. So this is something that then he talks about how, you have a lot of good resources out there where people are doing things like animations, cartoons, showing a lot of the processes, and, going beyond these abstract acronyms to these really, things that really help you understand what's going on in the biology. And so he kind of advocates for this idea of, how do we make this easier for people to understand coming in. I thought this article was very relevant to this group because that's what we kind of try to do in this group. One of the missions of the group is to make this stuff easier to understand. We're creating educational tools and analytical tools, and sometimes it's easy to lose sight of the fact that it's a lot of hard stuff, and this, I guess, holds for the computational stuff as well. If you don't really understand much about machine learning, sometimes these meetings can be kind of a slog in that sense too, because you have a lot of jargon thrown around. So, he mentions that people, a lot of drawings are good, a lot of illustrations are good. There's this book, A Computer Scientist's Guide to Cell Biology by William Cohen. And he mentions this book, and I think maybe if it's available online, I'm not sure, you might want to give it a read through if that's something that if you're a computer scientist, or even if you're not, and you only want to know cell biology, this might be a good way to kind of get a better appreciation for it. And so he talks a lot about people drawing with technologies. So, a lot of molecular biology even is based on illustrations and animations. And so those are very important parts of the learning process too. And so this last resource he mentions, The Eighth Day of Creation, Makers of the Revolution in Biology by Horace Judson. So this is another book that might be interesting for people so that they, they get a little bit better foundation in their biological thinking. So, so this is a pretty good article if you're interested in education and maybe like presenting your work more clearly. So there's that. And then Dick sent me, he's not here this week, but he sent me this paper on arrested coalescence in multicellular aggregates. So multicellular aggregates are known to exhibit liquid-like properties. So these are like a bunch of cells that are sort of in an aggregate. They're not a multicellular colony per se, but they're sort of these aggregates. And they exhibit liquid-like properties, so they look like a liquid in their behavior. The fusion process of two cell aggregates is commonly studied as the coalescence of two viscous drops. However, tissues are complex materials which usually exhibit viscoelastic behavior. It is known that elastic effects can prevent the complete fusion of two drops, a phenomenon known as arrested coalescence. Here we report the presence of this phenomenon in aggregates of mouse embryonic stem cells and provide a theoretical framework which agrees with the experiments. In addition, agent-based simulations show that a cell protrusion activity controls a solid to fluid phase transition. So this is where you have these sort of states of matter, and now we think of them in terms of physics, but now we're thinking about this existing with biological cells as well. Revealing that the arrested coalescence can be found in the vicinity of an unjamming transition. So this is where there's this field called soft materials, and one of the things that they found in materials is that when you have these granular materials, if they get to a certain density, they undergo this what they call a jamming phase transition, which means if you have a low density of particles, they move around and they're free to move around, and then at some density, they stop moving around and they jam together, and so that means they can't move all of a sudden. They're restricted in their movement. And this happens very quickly, at a certain density. And so what they're talking about is this sort of transition between jamming and unjamming. And so this is a really interesting sort of thing. So you can do this, you can analyze this with an agent-based model where you have a bunch of agents that are these cells. And I think we've shown these agent-based models in the group. So you know that you can add like cells at a certain density and they move around, and you can simulate this process, and you can actually observe these type of phase transitions. So by analyzing the dynamics of the fusion process, it is possible to infer mechanical parameters of the aggregates, such as viscoelastic reaction time and the elastocapillary number. So these are parameters of the physical nature of these cells. Our work provides a simple in vitro method to characterize the mechanical properties of 3D multicellular aggregates and sheds light on the impact of cellular activity on tissue mechanics. So this sounds like something that Susan would be very interested in. This is, really about the shaping process during morphogenesis, how organs are built. And so this is an example here. I don't know if I can zoom in on this, but you can see that they've got these cells. These are stem cells, and then they form these cell aggregates, and then they fuse together these aggregates, and this is what they call arrested coalescence. And so they can measure this. They can also simulate it with a agent-based model. They look at the different physical properties of these aggregates, and when they come together, so they're coalescing together, they're sort of almost like the two clusters are overlapping, whereas when they're not overlapping, there's no coalescence. And then, so then they get into the agent-based simulations. So they use a Kelvin-Voigt model, which is a physical model. They're trying to fit this to the experimental data, but then they don't really get enough information from that. They think they think that the material properties of the cell aggregates are much more complicated than we can get from a single physical model, and so we turn to these type of agent-based models. And so they're actually using a GPU to model this. I don't know if they show graphs of this. Well, here's one here, I guess. This is their agent-based simulation of these aggregates. So they have these red and blue cells. Each one of these cells is an agent, and they're watching it coalesce into, the two clusters coalesce into a single cluster. And then they have this regime here where they have protrusion strength and protrusion ratio, and they have this sort of map of the different phase regimes. So they have no coalescence, arrested coalescence, complete coalescence. And so you can build a graph like that where you have this transition. And this is, of course, the transition here, this dotted line. And so that's, if you want to read more about that, that paper's in the folder as well. And then I wanted to talk about two other things before we move on. These are short things. So the first one is this is a book that's old. if you're interested in old books or, just sort of the history of developmental biology, there's this book, Modern Theories of Development, from 1938. This is von Bertalanffy and Whittaker, who are two classic authors in this area. So this is, some of the views in this might be dated, but they kind of talk a lot about sort of the current theories of development. And this is sort of a record of this book. So I just found this on, in my readings. I thought that might be interesting to bring up some of the older classic books. This is one of the books. Again, this is almost 100 years old, so it's kind of, Modern Theories is kind of a bad name for it, maybe at this point. But, so they basically talk about, it is pointed out that a crisis has been reached in biology in 1938 due to the rapid accumulation of facts without clear theoretical laws. And so this is the state that they found themselves in 1938, and maybe today even. The controversy between mechanism and vitalism is revived, and is shown where each of these theories of life breaks down. The author also considers the physiochemical explanation of single phenomena in the organism, and that does not suffice for the foundation of a theoretical biology, as it fails to establish laws to explain the arrangement of organization of material processes. And so there's this idea, if you're interested in the history of theories, I know we have this theory building initiative that I've been talking about. It's really just a way to present this to people, and I think this will be part of that discussion where people are interested in how do you build a theory, because you can't really, once we interpret the data that we analyze, we build an analysis and then we present the results, then we really need a theory to interpret it. And so this is what this is part of. So I just wanted to bring this to your attention. Finally, I want to point to this this open training course. This has been around for a while, but I was reminded of it. Dr. Ken Ho, he used to be at Riken, and now he's moved to England. And he's been in the group from time to time in the past. I think he's spearheading this 4D cell nuclear tracking online course. And so what this is it's a course on cell tracking, where they track the nucleus. And in fact, a lot of the microscopy data that we work with in this group are based on these sort of cell tracking methods, where they usually stain the cell with some marker, and then they can track the cell in microscopy images, so they can get it into a sort of a common framework. So you can look at how cells move, but you're tracking the nucleus. So it's not the entire cell necessarily. A lot of times it's just the position of the nucleus, the time that it appears, the time that it divides. And so this is a course. It may be beyond, if you're interested just in the output data, it may be beyond what you're interested in. But it's basically goes through a lot of the stuff that, they use MATLAB to go through a lot of these analyses. So they introduce 4D cell tracking as a concept and cell dynamics analysis techniques. That doesn't assume you have any prior knowledge of programming. But it does allow you it introduces these ideas for image processing of 4D images, for labeling interest regions and then tracking them over time. And then this course introduces the use of this public database SSBD, which is something that we use. It's a very good resource for data, input data for models. There's a lot of data on different organisms for development or for other types of cell biology. And so if you're interested in that, maybe even if you're into machine learning, it'd be good to go through these materials to get a sort of an idea of how the data are collected. So it kind of goes back to our input data theme. So now, if you're Krishna, if you want to present, did you want to present?
Yes, presenting.
Okay.
Just give me 1 second.
Bradley, can you share something beginner friendly, like some topics I can't understand, like the new something you showed in the paper. So can you share that link?
Oh yeah, I can actually help. Are you on Slack?
Okay, okay.
I'll send
And
it.
some like something beginner friendly for the newcomers or for the computer scientists who
Yeah,
can understand the biology.
yeah. We actually.
Especially,
Yeah, I have some. I can send some resources. Yeah, we have a
yeah,
couple,
I
but
think
yeah.
Drop your email. I'll send you some.
Okay, yeah.
okay.
Okay, drop your email. I have some, some things that are beginner friendly.
Beginner.
So, is my screen visible?
Not yet.
No,
I guess
There
it
we
is
go.
now.
It's coming up. Yeah.
Yeah. Yeah. So, I want to present this. Yeah. So I've been working on this, you can say, problem on bioinformatics. So here, what I am doing, I am going to, do the PCA of the genomic data to, estimate the ancestry. So first of all, I'll tell you that what is Thousand Genomes Project. It was a project that came up in 2007, where at least 1,000 volunteers of when a single ethnicity were considered and their genomic data was taken. And here is an example of PCA, that's principal component analysis. So what I am going to do is, I guess this shows us all. So you can see this. This is the PCA of thousands genome data, and I'm going to merge it with my sample, the person whose ethnicity that I want to consider. And then, for example, if I whenever I visualize that data, I can estimate the ancestry of the person. So one second. So this is an example of genomic data. It's in VCA format. That is a variant called, one second, I guess I had a good picture. Okay. So what here happens is, first of all, the data that is I'm having is merged with the 1,000 genome data, and then after that I used in a tool named plink. It gives me the eigenvalues, and after that these eigenvalues are plotted in the language R to get the visualization. So for example, if I'm I'm having Asian people, and here I'm having African people, and my point is someone here. So I get the chance that the person whose ancestry I'm going to check is belongs somewhat close to the African people. And how it can be helpful, it is can be used in selective medication, because it is being found that different races respond to different medicines in a different way. There are some sorts of antibiotic that are said to be not as efficient in some races that they are in other. And one more thing is that early disease forecast. As you know that Indian people are more prone to diabetes, and there are different yeah, and like Caucasian people are more prone to pollen allergy, I guess. So what we can do is that if we are able to predict the ancestry of a person, we can check out that which type of disease he is more prone into. So these are just the things that I require: the variant call format of 1,000 genome data, the variant calling format of my own data. Then I know need a genome pen file, and we know need a reference genome file, and it is publicly available by the National Center of Biology Biotechnology information. So here is the data flow. First of all, data is collected, then it's converted in a format that is accessible by the tool plink, and then we exclude the variants that are not necessary. And after pruning each of the chromosomes, we get the eigenvalues. And plink only give us the eigenvalue. It doesn't visualize that chart. So that is to be done in the R language. So the things required for that, we need to have batch scripting for creating the scripts. We need plink. We need VCF tools. It is a Linux tool that is used to convert the VCF file into the BCF files, and we might need a little of Perl scripting so that we can call the libraries which will help us in the pruning of data. And we have C libraries, SAM tool, BCF tool, and HTS lib. So I guess that's all, and that's an example of how the VCF data looks. It can be opened into Microsoft Excel, so that to visualize it and see how it is. I guess that's all. Any questions? This is an example of PCA, but it is PCA of 1000 genome data. It doesn't have any other data. For example, like I have to merge my data in it to get the ancestry of my data. It is just the normal regular 1000 genome data. And you can see here reds denote African population, green denotes East Asians, and so on.
Yeah.
That's it.
Well, that's good. Thank you. I know this is the stuff you were talking about on Slack.
Yeah. I was badly stuck on it for a couple of weeks.
Yeah. And you're able to figure it out.
Yeah, actually, there was some error in conversion of files.
Yeah.
I was not able to convert my files, and it was, it really took me a couple of weeks to do it.
That's always a big sticking point in bioinformatics, is like file conversion and all that. So.
Yeah. We have a lot of formats. We have fast to BCF, and it's one tool works with one format, one doesn't work with one, and the debugging process was, very hectic.
Yeah.
So, any questions regarding my project?
Yeah, your project was very interesting, and
Thank you.
Yeah, just send a link to the Slack channel, then I'll check it out.
Yeah, sure.
And Bradley, I had another question. That is
okay.
I showed you the GANs thing, right?
Yeah.
So I had added a tutorial on GANs in the DevoWorm datasets demo repository, right?
Okay. Yeah.
So is it okay if I add another tutorial on conditional GANs?
Yeah.
This is something else, right? So I guess this will be helpful to someone who is starting out, because there's not a lot of information available on this.
Yeah, it'd be great. Yeah. Did you want to push it to the repository?
Yeah.
Okay, yeah, go ahead.
Then I'll create a beginner-friendly notebook which will have the markdown and the explanations and stuff. So, that's something I wanted to do. Is that okay?
Oh, yeah, that's good. That's good. Yeah.
Yeah, okay.
Look forward to it. Okay, well, thank you for attending. And if you have any questions, contact us on Slack or on email or whatever. And see you next week. Thanks.
One thing I want to ask Bradley.
All right.
It was regarding that rock wire. If it's the right time, you can explain. Otherwise, we can do it some other time.
Oh, we can probably do it offline. I can talk about it on Slack. So. Okay.
Yeah.
All right.
Yeah.
All right.
Bye. Nice.
Bye. See you next week.